Hello and welcome to this short video on observing and studying the human brain. My name is Professor Phil Newton, and what I hope you're able to do by the end of this short video is just to be able to describe some common methods for observing activity in the human brain. We're going to talk about methods that we use clinically and also methods that we use to try and research the human brain in an effort to understand how structure links to function. Here's a list of the methods that we're going to cover in this video. We're going to talk about the lesion-deficit model and then make our way through CT, PET, fMRI, and EEG. We're going to talk about how these are used clinically, how they're used in neuroscience research, and some of the main strengths and limitations of the different models. We're going to start then with the lesion-deficit model. This is perhaps the oldest way of studying the brain. The word lesion here simply means any sort of defined damage to a particular part of the brain. Obviously, in life, some people unfortunately experience illness or injury which results in damage to parts of their brain. If this damage also results in changes to their behavior or cognition or function, for example, then that gives us some evidence to link those damaged regions to those behaviors or cognitive processes which have then become impaired. One of the first and most famous examples of the knowledge advancement which can come from such an approach is this chap, Phineas Gage. Phineas worked on the railway in the mid-19th century in the USA. On one unfortunate day in 1848, Phineas had a very serious accident. What he's holding there in his left hand is an iron bar with a sharp and tapered end. An explosion caused this iron bar to shoot up through the side of his face and out through the top of his head, landing several feet away and causing serious damage to his prefrontal cortex in the left hemisphere. This damage was then worsened by infections during the recovery process. Ultimately, though, Phineas largely recovered the physical damage from the accident, but his personality had changed. He became impulsive and rude, and this is part of how we came to understand the role of the prefrontal cortex in executive function. There are many advantages to this sort of approach. The first is that it is a direct study of the consequences of damage or loss of a particular part of the brain on function, cognition, and behavior. The data from these sorts of studies are real and natural. They don't require additional extrapolation from physiological measurements in the way that some of the later methods we're going to talk about need to. There also exists the opportunity for pre and post measurements of the person's behavior, structure, or function. This particularly in the case where damage happens as a result of some planned or controlled surgery. One of the most famous examples of this is an individual called patient HM, later revealed to be a chap called Henry Moliason. He had intractable, incapacitating epilepsy, for whom the only treatment option back in 1953 was the surgical removal of his medial temporal lobes. Upon recovery from the surgery, he was not able to form new explicit memories, something he had not struggled with before the surgery. And so this told us that the medial temporal lobes are important for memory formation. This approach, though, is also associated with many limitations. One obvious one of these is the variability between patients and between injuries. One patient's injury is not going to be the same as another's, and then there's variability between the patients themselves. Very many different demographic characteristics can profoundly affect the function, structure, and cognitive processes associated with different parts of the brain. Then there are also some temporal challenges. The brain is not very good at repairing itself, but it does undergo some regain of function after damage. So studying a patient soon after an injury or a surgery might give you different results than if they were being studied a few months or even years later. Then there are some obvious ethical challenges with these sorts of approaches. The sorts of patients we're talking about here have undergone very serious injury or surgery and then had to go through a convalescent and recovery period. The last thing they might want is a neurologist, a neuroscientist, or psychiatrist talking to them about their behavior and their personality and asking questions and studying them. It might also not be in their best interest to be part of these scientific studies. Some of these limitations can be addressed by using animal models, but these then come with their own sets of ethical challenges. And of course, there's the validity challenge of mapping from animal studies to human. The case of Phineas Gage is an example of many of the strengths and limitations of this approach. We don't have great information on Gage's personality before his accident, and we don't really have great data on what happened to his personality afterwards. Many textbooks and accounts have exaggerated or mistaken the changes in his personality which took place as a result of the accident. It seems that the changes to Gage's personality did show some temporal features, getting better over time and then perhaps worse again in later years, although this may also just have been a result of aging or some further complications. Okay, so that's lesion deficit. Now we're going to go on to talk about computerized tomography, more commonly known as CT. CT is basically just a series of X-rays. And here you can see a series of just such a set of X-rays. What's very obvious is that bone shows up very well on X-ray. Blood also shows up very well on X-ray. The soft brain tissue, although some structural features are readily available, doesn't have quite the same resolution. However, this series of X-rays can be assembled together into a three-dimensional format, giving a fairly quick and easy account of the structure of an individual's brain at a particular time. The strengths of CT are that it's very quick. It's particularly useful then for acute trauma. Its high resolution, particularly for structures like bone and blood, makes it very useful in trying to understand what's gone on when somebody suffered an acute head injury or a stroke. This is particularly helped by its great visualization of bone and blood, which are clearly key to understanding what's happened when someone's had acute brain injury. And the 2D images can be reconstructed into a 3D format. Some of the limitations, though, of CT, first is that it uses ionizing radiation. It's an X-ray. This means that the patient is exposed to something which, if exposed in significant amounts, could result in damage to their health. As we said earlier, it's not great for soft tissue, and the brain is pretty soft, and it provides only a snapshot in time. It's not a great account of an individual's change in function in a rapid period. You couldn't put someone in a CT scan and have them perform some sort of cognitive task and look for changes in brain function acutely, for example. Next, we're going to talk about positron emission tomography, otherwise known as PET. This involves the monitoring of a radioactive tracer. The patient is fed the radioactive tracer or consumes it in some way. It then makes its way around the brain, and normally it's something that either binds or is metabolized, and the path of that tracer around the brain can be monitored over a period of time, although the resulting images are normally just a snapshot in time. Here then are some common examples of the way in which PET can be used. This is an image from a study where patients were fed radiolabeled glucose, which is a very common way of using PET imaging. On the left-hand side, you can then see where that glucose goes and where and how it is metabolized in the PET scan from a patient without Alzheimer's disease. And on the right-hand side, you can see a PET scan from a patient with Alzheimer's disease. What this very simply shows is different levels of metabolic activity in patients with and without Alzheimer's disease. We can also use much more specific radiolabeled tracers. I'm going to show you an example of studies using Pittsburgh compound B. This is a compound which binds the fibrillar version of beta amyloid. Beta amyloid, the fibrillar version of it at least, is a principal component in the plaques that are seen in patients with Alzheimer's disease. On the left-hand side, then, you can see now a cognitively healthy person, and on the right-hand side, a patient with Alzheimer's disease. This time, the patient with Alzheimer's disease has their brain lit up by the radioactive tracer because it's binding to the plaques that are characteristic of Alzheimer's disease. PET has a number of strengths as an approach. It can be very specific if we're using a very specific radiolabeled tracer. We can measure absolute levels of activity using PET rather than relative levels as we do in other methods. And it can be combined with CT to get the relative strengths of both of those methods together in one set of images. There are some limitations to PET, however. Again, it uses ionizing radiation, and it can be very expensive, particularly the tracer itself. PET only has a modest spatial resolution, and you saw that on the images that I showed you. They're quite blurry, even though they do give us gross aggregate differences. And again, it only provides a snapshot in time. We're now going to move on to talk about functional magnetic resonance imaging, more commonly known as fMRI. The underlying physics of how fMRI works are quite complex and something we probably need to cover another day in a different video. Very simply, what fMRI measures is the BOLD signal, where BOLD stands for blood oxygen level dependent. The BOLD signal is generated on the basis that hemoglobin has different magnetic properties when it has bound oxygen versus when it has not bound oxygen. And this difference in the magnetic properties can be detected using a magnet. Different levels then of oxygenated versus deoxygenated hemoglobin are thought to be indicative of different levels of activity in a particular part of the brain. When a part of the brain becomes more active, it uses more oxygen, and so there will be a greater concentration of oxygenated hemoglobin sent to that part of the brain. We need to spend a second considering the unit of measurement of fMRI, which is a three-dimensional measurement system. The unit of measurement of fMRI is the voxel, which is basically a three-dimensional pixel. Pixels are a two-dimensional feature of images. Here is a two-dimensional image, and you can see if we zoom in that it is formed of pixels, little squares of different colors and shades, and when these are aggregated together at a distance, we're able to, using our own visual cortex, comprise an image. A pixel then is a two-dimensional square. A voxel is basically just a three-dimensional version of a pixel. This is important because voxel size is an important feature of different types and strengths of fMRI. Where we have a voxel that is 3 millimeters cubed, which is very common, particularly in many clinical applications of fMRI, then we have a voxel that contains 2 million neurons. Remember from other videos that for every neuron there is more or less a non-neuronal cell, so a 3 millimeter voxel will have 2 million glial cells in it as well. This is important to bear in mind. Even though 3 millimeters is a very small area of the brain, it's got 4 million cells in it. So we're not looking at the activity of individual or even really groups of cells. We're looking at gross activation of a particular part of the brain. Using a stronger magnet allows us to apply smaller voxel sizes when we're analyzing our data. Even a 1 millimeter voxel, though, has got 72,000 neurons in it and 72,000 glial cells. This is still a lot of cells, but it is considerably fewer than the 3 millimeter voxel. This is an image then from some fairly old fMRI studies. I'll give you a reference at the end to the paper that this comes from. And this fMRI is showing you activation of the basal ganglia in a particular clinical situation where in the bottom, the basal ganglia are thought to be less active. You can see that we've got pretty good resolution, pretty good illustration of the basal ganglia, and the color coding of the image allows us to identify relative levels of activity. The strength then of fMRI is that it is functional in real time. Patients can be placed in a scanner and they can be asked to do a particular cognitive task, and the changes in the oxygen utilization of particular parts of the brain can be monitored in real time. So it's got some temporal capacity in a way that PET and CT do not. It shows pretty good spatial resolution compared to many other imaging formats, particularly with the higher strength magnets allowing us to reduce the voxel size. And it doesn't use any radiation, which means that it is less damaging or less hazardous for the patients or the subjects who are part of these studies. There are many limitations, though, to fMRI. It has pretty poor temporal resolution. It takes quite a long time for the blood oxygen levels to go up in the particular region of the brain that's becoming active, whereas cognition happens on the order of milliseconds. The changes in blood oxygen happen over the order of seconds, 10 to 20 seconds, for example. The subject then is very restricted when undergoing the scan, and the environment in which they're undergoing that scan can be very aversive. This is a picture of a typical fMRI scanner. Many of you may have been in one of these scanning machines yourself, and you'll know that you get slid in to that circular structure there, which is where the magnet rotates around the patient. It's very loud, it's very confined, and patients may have to be in there, or subjects may have to be in there for some considerable time when undergoing a cognitive task. This can obviously produce changes in cognition, changes in anxiety and stress, which may then confound the results. The final method that we're going to talk about is EEG, more fully known as electroencephalography. EEG is interesting because it's one of the oldest ways of studying the brain compared to PET, CT, and fMRI, and yet it is one of the most poorly understood, in part because it measures something slightly different to those other imaging modalities. EEG then involves a series of electrodes being placed across the scalp of the person who is undergoing the EEG. You can see here a color-coded representation of where the electrodes might be placed and how that maps on then to the different cortical regions that we've covered in another video. Referenced electrodes are then placed, for example, on the ear or on the nose, or regions close to those. The electrical activity then of groups of neurons in a particular region of the cortex can be picked up by the different electrodes and compared to the reference electrodes from which no changes in electrical activity are being measured. It's important to be clear of what EEG is measuring. It's electrical activity in the cerebral cortex and it's groups of neurons firing together, but it's not really measuring action potentials. It's more measuring the consequences of action potentials. A neuron becomes polarized during the firing of an action potential. Parts of it are negative and parts of it are positive, and the difference between those is what is being picked up by EEG, and then the difference picked up by one particular electrode on a cortical region of the scalp versus a reference electrode where there shouldn't be as much electrical charge. It also measures groups of neurons firing together. It doesn't measure the activity of individual neurons because the charge difference generated is too small. Because it measures groups of neurons firing together, what we see on EEG are characteristic waves. Groups of neurons we now know fire in these waves, in these neuronal oscillations. The meaning of these oscillations is still the subject of intense debate and scrutiny, but it's clear that they're there and that they can be correlated to certain types of behavior and certain types of cognition. The key feature of EEG then is that it measures a temporal rather than a spatial feature of brain activity. CT, PET, and fMRI tell us whereabouts in the brain spatially has become active or has become damaged when we scan a particular patient or subject. EEG is measuring changes over time much more than some of those other imaging modalities. Its spatial characteristics are not that great, but its temporal characteristics are much better because it's able to record changes in brain activity on the order of milliseconds. Let me show you some of these characteristic waves that we can see using EEG. Here is a typical beta wave, recorded when someone is awake and cognitively active. The scale on the bottom of the wave there is in seconds, so that wave represents a second, and you can see the cycling of the wave corresponds to approximately 13 to 30 hertz. That means there's approximately 13 to 30 waves over the course of a second. Here then is a delta wave. You can see that this is much slower. This is basically a 1 hertz wave. There's one wave over the course of the second, and this is the sort of wave that we might see when someone is engaged in deep sleep. Here then is a gamma wave. This is the fastest firing, if you like, or the highest rate of oscillation. You can see here that the hertz rating of a gamma wave is between 30 and over 100. Gamma waves are the sorts of waves that we would see in a particular region of the brain when someone is engaging in very cognitively demanding tasks. Gamma waves are often lost in conditions such as Alzheimer's disease from certain parts of the cortex. EEG has a number of strengths. It has a great temporal resolution. As I showed you on the previous slide, we're recording data on the order of milliseconds, which is something that's just not possible with any of the other methods. It's non-invasive. A person has to wear a scalp cap or to wear a whole bunch of electrodes, but there's no great big scanner involved and there's no invasive surgery. It's also then pretty cheap to do as a result of that. It does also, however, have some limitations. It has pretty poor spatial resolution, basically limited to the number of electrodes that can be fitted, and obviously, as I mentioned earlier, it only records activity really from the cortex, although it is possible to pick up signals from elsewhere. There are a number of artifacts that can be also picked up during EEG measurements. Electrical activity is not something that's just confined to the brain. Other regions of the body and other structures in the body, for example muscles, are electrically powered, and that will generate signals that can be picked up by EEG. And this has to be analyzed using fairly sophisticated analysis, which is beyond most of us. Okay, then let's just finish with a summary of the main methods that we've covered in this video. These are the main methods for studying and observing activity in the human brain. We started with the lesion-deficit model and some famous examples such as Phineas Gage and patient HM. The lesion-deficit model simply allows us to look for changes in cognition, behavior, and function in patients who've unfortunately experienced damage to a particular part of the brain. CT scans are basically a series of X-rays. They're quick and fairly cheap, and they're great for identifying the nature and extent of acute trauma in situations such as head injury and stroke, but they're not great at imaging the soft tissue of the brain. PET scans allow us to follow the course of a radioactive tracer once the patient has been given that radioactive tracer to ingest. The radioactive tracer can be very specific from a molecular and chemical basis, meaning it can allow us to identify very specific molecular and chemical activities and processes in the brain. Radiolabeled glucose, for example, can be used to look for changes in metabolic activity, or chemicals that bind the plaques commonly associated with Alzheimer's disease can allow us to see the extent of those plaques in patient brains in a non-invasive way. fMRI allows us to monitor changes in oxygen usage in particular regions of the brain. Increased oxygen usage is associated with an increase in the activity in that particular region of the brain, while the F in fMRI stands for functional, which tells us that fMRI allows us to monitor real-time changes in the activity of parts of the brain while a patient undergoes a particular cognitive task. Developments in the technology mean we're now able to do this at a pretty good spatial resolution, i.e. we can zoom in on particular parts of the brain while a patient performs a task. The changes in activity we can measure with fMRI are quite slow when compared to the speed with which the human brain functions, and the environment in which the participant is being scanned can be quite aversive. But fMRI is probably the method that's giving us the most rapid advances in our understanding in the structure and function of the human brain at the moment. One striking example of this is recent studies showing the ability of sophisticated AI models to decode fMRI data. Studies have recorded fMRI data from participants when those participants were viewing a set of images, for example, giraffes and fire engines. The AI was then given the fMRI data and asked to determine what it was that the participant was looking at when the fMRI data were recorded, and the AI does a pretty good job. It can say, yes, this participant was looking at a fire engine or a giraffe, for example. EEG is a long-established method of measuring synchronous firing of groups of cortical neurons. It provides a very different view of brain activity, allowing us to see very rapid changes in neuronal oscillations. All of these approaches complement each other to some extent and can be used in combination. For example, high-quality imaging of patients with damage to a part of the brain can, when combined with neurological exam and detailed observation of the behavior and cognition of that patient, provide us with a much more detailed view of the role of the particular brain region in a certain type of neurological function. It also allows us then to move beyond lesion and deficit to what we might call difference and difference. We can look for patterns of difference in the structure or functional activity of certain regions of the brain of healthy individuals and combine this with behavior and other observations, again to tell us what a particular brain region is doing in regards to a particular type of cognition or function. Okay, that's it. This is the image credits. As always, the images are all open source images from Wikimedia Commons, and they're all used under an open source license for teaching. Any questions, please do leave some comments or send me an email. That's my email address right there, and I'll see you in another video. Bye-bye.